Welcome back. We are going to continue our discussion on chromosomes and location of genes by talking today about Thomas Hunt Morgan and his fruit fly experiments. And Morgan is really given the credit for discovering the location, giving us evidence for what Mendel was saying about these heritable factors and the fact that these genes, these alleles, have to be located on specific places on the chromosome. And Morgan decides to work with fruit flies, which are a really nice model organism for a lot of genetic studies. In fact, it's a pretty common freshman biology college lab to do these fruit fly experiments because the fruit flies breed at a really high rate, so they make lots of offspring every breeding event. They're sexually mature and ready to make a new generation about every 2 weeks, so they reproduce time-wise very quickly. And genetically, they're not overly complex organisms. They've got 4 pairs of homologous chromosomes, so not a whole lot of genetic information, but enough to kind of do what most genetic studies, at least in the early stages, are looking for. The other nice thing about fruit flies is that Morgan noticed that there is very often a wild or a normal phenotype, so the physical expression of genes, that have a common alternative or a mutant phenotype to it as well. So one of the examples is their eye color. Okay, and Morgan was looking at the mutant trait versus the wild trait and distinguishing how often these differences showed up. So one of the things you should be familiar with when we see these written out in terms of a genotype is that it's typically given the wild is given a plus sign, and the actual letters that are associated with that are associated with the mutant type. So here's my mutant type, the white eye. So WW without the subscript tells you that it is the mutant, and generally these mutants are recessive. So they would have to be homozygous in order to show up. The red eyes is the wild or the normal phenotype, what most of the flies are going to look like. And so we're going to give it a W plus to say that it's a wild. And in this case, this one's written out as a homozygous dominant for that wild trait. One of Morgan's early experiments was to take a male fly who was a mutant, so white eyes, and mate it with a female who was red eyes or wild type. And what he found in that first generation, or that F1 first filial generation, is that 100% of those offspring all looked mutant, all looked wild, they all have red eyes. He then took 2 individuals from that first filial generation, mated them together, and looked at their offspring. And that F2 generation, he got that normal Mendelian 3-to-1 ratio, but what he noticed was all the mutants, so that 1 and 3 with the white eyes, they were all male. And that was able to give Morgan convincing evidence that, all right, if all of the males have to show that mutant, we know that males are XY, females are XX, then that trait has to be located on the X chromosome because the males don't have a second X to kind of cover up or act as a carrier. The females, however, are all going to be carriers. Okay? Or at least half of them will be. And so Morgan's discovery of this transmission is the convincing evidence that chromosomes are correlated to certain traits. Okay, that specific traits are associated with specific chromosomes. And so from there, he starts taking a look at how those alleles are actually located on the chromosome itself. So once we get to the idea that yes, in fact, there are specific genes being carried by specific chromosomes, then he started looking for where those genes are actually located. And what he came to find is that some genes are actually linked together, or what we call linked genes. And linked genes mean that those genes don't get separated out when we go through crossing over. All right? So if we remember, crossing over happens during prophase 1 in meiosis, where we take homologous chromosomes and we take a little piece of the chromosome that that organism got from its mother and mix it up with the chromosome from the father, so that you end up with these parental types from the chromatids that didn't cross over and the recombinant types that did. All right, and what Morgan ends up seeing is that if we go through normal meiosis, we should expect a 50-50. We should expect 2 gametes to be parental and 2 gametes to be recombinant. That's not always the case, depending on where those genes are located on the chromosome itself. All right, and so those linked genes don't follow this 50-50. So what Morgan sees is that genes that are located really close together on the same chromosome end up being linked, meaning that they don't end up getting assorted independently. They don't get crossed over separately. Your unlinked genes, however, are far enough apart on the chromosome that when we go through crossing over, they have an equal likelihood to get assorted independently or get crossed over separately. So how do we figure this out? Well, we have to look at the number of recombinant offspring we get from a cross versus how many are parental. Okay, and remember that those recombinant offspring are the ones who have the new set of combination of traits. So they've got some mixture of both of the parental traits. If this is a normal, non-linked situation, you should get 50-50. 50% should be recombinant, the other 50 should be parental. Okay, and if you think back to Mendel's P's, that's why we can do those big Punnett squares, because we're assuming those traits are not linked together. So Morgan looks at the appearance of these recombinant phenotypes in the offspring of certain traits. And what he noticed was that he wasn't always getting that 50-50 ratio of recombinant versus parental. And so he comes to understand that the actual breaking of the connection between genes, the crossing over, is in fact this mechanism that is causing some genes to get crossed over, creating those recombinants, while some genes are not getting crossed over because of where they are located on the chromosome. All right. And so again, remember that the crossing over happens as a way to increase genetic variation in prophase 1 of meiosis. And the likelihood of the crossover really depends on how closely the genes are located to each other. So if they are physically close together on that same chromosome, it's going to decrease the chance of them crossing over. Not that it can never happen, but it's far less likely. The further apart they are from each other, the closer we should see that 50-50 parental versus recombinant, and the more likely that they are to get crossed over separately. So here's sort of a scenario that we're looking at. These genes are physically located very close together on the same chromosome. So when they go through crossing over, all right, those alleles tend to all stay together, and as a result, we get a lot more of the parental types. This is parental, it looks exactly like the parent. This looks like parental again, exactly like the parent. This is also parental, it looks like the parent. This is also parental. So in this case, we, as a very simplistic number, get 100% parental, and we don't have any recombinant at all because those genes are so close to each other. If I take this scenario on this side, however, now I've got alleles that are very far apart on the chromosome. We go through crossing over. Well, here's a parental type. It looks just like the parent. Here's the other parental type. Looks just like the parent chromosome. But these two here in the middle are a mixture of those two genes. So here's the one from the red chromosome, here's the one from the blue, and vice versa. These are your recombinants. In this case, I've got a very traditional Mendelian cross of 50% parental and 50% recombinant. Now you can have things anywhere in between. So the further apart two genes are, the higher the probability that the crossover happens, and so the higher the recombinant frequency. Recombinant frequency, we're just talking about a mathematical representation. It's the percentage of recombinant organisms who show the mixture of the parent traits in the total pool of offspring that you create. All right, so a linked gene has less than 50% of that recombinance. A non-linked gene will have 50% or more. So the proportion of your gametes, your sex cells, sperm or egg, that have these recombinant alleles then allows us to actually map where the genes are located on the chromosome itself. We can create a genetic map of alleles on a chromosome based on that recombinant frequency that we see in the offspring. Again, the further apart the genes are on the chromosome, the more likely they are to separate during crossing over, the higher the recombinant frequency, and that starts allowing us to put numbers to where those are. So we can do this by actual number, or we can do it by percentage. I think the percentage sometimes ends up being a little easier. So the proportion of your gametes with recombinant alleles allows us to now actually put a number to utilize to create a map of where these alleles are located on genes. For every percent of recombinant frequency that we see, we can talk about it as a single map unit. Your chromosomes here's a chromosome length, right? And this allele B and this allele VG recombinate 17% of the time. So there's 17 map units between them. B and CN recombinate 9% of the time. So there's 9% or 9 map units between them, and 9.5 between CN and VG. So knowing all of that information, you can start figuring out where they're located in relation to each other. And if we add 9 plus 9.5, you get approximately 17%. Okay? So that's one way that we can start doing this, and we're going to do some practice problems over the next couple of days of how this actually works.